We go to the first compartment, which is the mouth, and there we have the teeth, which are for us important for development of tooth caries. Sugar intake is now more or less limited to 10% of the total energy intake, and this is mainly based on potential role that sugar can play in development of tooth caries in humans. How can they do that? It's by making biofilms. The biofilms will normally deposit here on the lamelles, and as a result of that, it will attract bacteria. And these bacteria will acidify the medium, and then these acidifications will normally attack the tooth and leading to tooth caries. And so a lot of research is now going on to try and find a way how we can destabilize biofilms. The best way, of course, is by cleaning your teeth. That is quite simple every day. But there's also research going on for toothpaste, for example, to see how we can limit the development of dental plaques and as such the development of tooth caries. The treatment is oral hygiene, that is clear, and in the food industry, a lot of attention now is paid to sugar replacers, and where sugar is replaced by artificial sweeteners, which probably are also not that healthy as we were thinking, because they also might have negative effects. I will come back to that later. But we're also working more and more on so-called natural sugars. So these are not classical sugars, but new types of sugars with new structures that look a little bit like natural sugars, but that do not lead to tooth decay. So they are not that well recognizable by microorganisms. They're not that well capable of producing a biofilm, and they will not acidify the medium. And as such, they may be useful for overall dental health. Some of them are, for example, sugar alcohols that you might already heard about. You can find them in the jams and in light products, which are already on the market. But we also have specific types of natural sugars that appear in nature, but that have another bond. And this other bond makes it more difficult to break it down. So in this situation, it will not acidify the medium. I think if we talk about sugars, I will give you some further examples. Another important role and function of digestion in the mouth is taste. And so, as you know, if people don't like eating a certain food, if the taste is not well, they will usually not consume it. And so, in overall human health, if you want to see that people eat more healthy, at the same time, you have to see that the food is a little bit tasty and that people like consuming it. In order to understand that better, there's a lot of research going on in the area of taste receptors. So try to understand how taste is developing in humans. And we have 5 tastes, which is called the umami taste, the sweet taste, the bitter taste, the salty taste, and the acid taste. And for all of them, we have a receptor, which is located in the membrane. And this is found more or less on the tongue surface. And you can see here on the bottom all the different compounds that can interact with the receptor. L-glutamate, for example, nucleotides will interact for the umami taste, which is a taste that is more classically found in Asian countries. In Europe, umami is a little bit less applicable and used. The second one is much more popular. It's the sweet taste with a clearly identified receptor. In this case, sugars can bound, artificial sweeteners, amino acids, and all types of other types of proteins even can taste sweet if they bound to the right receptors. The bitter taste is more simplified in molecular structure, but a whole set of compounds can interact with it. And bitter taste is usually developed throughout time in order to detract food, to warn people that you consume something that potentially could be toxic. And then for the salty taste, there is a small channel here where only the sodium element can interact. So this is a very small atom. It can actually go through this channel, and then in the end, it can actually cause a salty taste. Salt is nowadays negative because it can have a negative effect on human health, and sodium is replaced, for example, by potassium. Potassium is more healthy, but doesn't have the right taste because potassium is bigger than sodium and it cannot pass through the channel. So as a result, the salty taste coming from potassium is a little bit less than what you have with sodium. And then for the salty taste, we have, for example, carbonated drinks. The carbonate is converted into bicarbonate and protons, and the protons can act on the receptor. So it's a mild acidification, and for most organic acids, this is the case for all types of foods that give you a slightly acidified medium. The very strong acids are usually detrimental. And so these ones we do not want because they can actually damage the epithelium. You might think that taste is only relevant for the mouth and for the tongue. This is actually wrong because nowadays we know that taste receptors are present in the whole GI tract. So also present, for example, in the intestine. And this means that you can find there sweet receptors, umami receptors, bitter receptors, and even receptors for lipids, which are not well characterized yet, but we know that they are physically there. The question could be, why are they there? Well, we know that if a compound bounds with a sweet receptor, there is a process inside the epithelial cell that finally leads to the production and the interaction with the brain system, so that you can take up more foods or more nutrients, and that you can actually metabolize it in the human body. So there's a whole pathway behind it, and these pathways could play a role in several physiological functions in the human body. For example, for food intake, for immune functions, for the uptake of glucose, for transit time development, and for insulin resistance. So in other words, taste perception in the intestine could play a role in designing a specific transit time in the GI tract. So the body is actually tasting what is physically there, and based on that, it will respond by speeding up the food or by slowing down the transit time so that the digestion process can be improved. And so there's a mutual interaction between what is present in the GI tract and what the human body is actually going to do. Please remember that, because in some of the problem-based learning, we might use that to understand how taste can actually influence the whole body metabolism, not only taste perception as such, but the whole metabolism in the human body. So if we know that taste is important, if we know the receptors and we can characterize them, we can also understand their activities, then of course a very important question is, how does taste develop? What can we actually do to change taste perceptions in humans? And here I have an experiment, and you can find the article on the website in case you would like to read it. It's a very simple approach to illustrate the importance of taste development. We have 3 intervention groups. The WW is a reference, and the WC is a group where intervention was done during lactation, so when mothers were giving breast milk. While the CW is a group where the intervention is done during pregnancy. So you interfere during pregnancy, or you interfere during lactation, or you don't interfere at all, which is then the control group. So what did they actually do? They asked mothers during pregnancy, for the black bars, they asked them to eat a lot of bitter vegetables, like, for example, cabbage, during pregnancy. So during the whole 9 months of pregnancy, those women were consuming bitter taste foods, especially cabbages, throughout the week. I think 2 or 3 times in a week they were just eating cabbages. For the second intervention group, you do the same, but not during pregnancy, but during lactation. So after the child is born and it gets breastfed, during those months of breastfeeding, the mother is consuming a lot of bitter type foods, like cabbages. And for the third group, this is the control group, they were not eating bitter foods, they were eating other types of foods in combination with classical water as a placebo. At the end, when the child is born and we are at the end of the lactation period, it means that the child will receive for the first time solid foods, and we call that the weaning phase. So next to breast milk, the child receives also solid foods. In this experiment, we're going to give the child solid foods that are rich in the same bitter vegetables. So for example, a porridge which is made of cabbage. And we're going to measure how the child will respond. And we do that in 3 ways. The first one is to look at the child's faces. So if you have a negative face, then it means that the child doesn't like it. The second one is the mother's perception. A mother knows her child very well, so she can understand whether the child likes it or not. And the third one is the real intake, which you can also measure by a couple of techniques. So what did we see here? We saw that if you give the cabbages to the mother when she's pregnant, that the child has the lowest negative face, the mother has the highest perception, and the child has the highest intake. If you do the same experiment with lactation, it has the same trend, but it's a little bit less significant compared to what you have with pregnancy. And compared to the control samples, you see a very clear difference. So what can we learn from this experiment? We learn that taste is developing very early in life. And the behavior of the mother during pregnancy can already determine the behavior of the child when it's 6 months old or older. Yeah, so it's crucially important that you see the vulnerable periods of life, which is pregnancy in combination with lactation, because all the elements to which the mother is exposed at that period of time can also influence the development of the child at later age. And if we want to interfere also with taste development, the earlier you can do that, the better. That is actually what the conclusion is here. What I don't explain to you yet is the mechanism. So how is it possible that at a very young age, people are more vulnerable than at older ages? In chapter 5, when we are discussing about different types of theories, also genetic theories, I will explain that to you a little bit further. But you can already think now that the DNA and the genetic material is more vulnerable the more earlier you interfere in life. The older you get, the less impact it can have. And think about the language. If I would be very, very young, it would be probably easy for me to learn Chinese. But if I'm older and I have grown up, for example, in a European community, learn Chinese when I'm 30, 40 years is much more difficult. Yeah, it's a little bit the same idea. The younger, the larger the impact, the more easier it is to achieve changes in the metabolism. Then we have the stomach. The stomach has a wall. What is just important for you to know is that the epithelium of the wall is organic material, and this is very sensitive for breakdown. So if I'm working in the lumen of the stomach, this is very acid, pH 2. So pH 2 will just break down the wall. And in order to avoid that, I need a lot of mucus, which will actually protect the upper side of the wall. How can I produce that? Well, I can produce that by using glands. So here you have all these glands that are located inside the wall of the stomach. And you have different types of cells, like the parietal cells, we have the chief cells, we have the enteroendocrine cells. So they actually produce mucus that can be released into the medium and that can protect the cells. We also have chief cells that are producing a lot of enzymes that can be released for digestion. And we have enteroendocrine cells that are actually having hormonal activity. And we also have here the parietal cells that are mainly also releasing acids in the environment. So the globulet cells that are physically there are producing mucins to protect the cells layers. The chief cells are producing pepsin and lipases, which are enzymes that can be used in the digestion process. You will see later what they actually do. Pepsin is used for breakdown of proteins. Lipase is used for breaking down lipids. The parietal cells are producing acids, also an intrinsic factor, which is actually protecting vitamin B12, because B12 is not acid tolerant. So when you consume B12, it comes into the stomach. The pH is so low that it can break down B12. And to avoid that, there is an interaction between B12 and intrinsic factor, which is a sort of a protein, and it can protect the transit through the stomach. And in the small intestine, you then have the absorption of B12. The enteroendocrine cells, they produce hormones, in this case gastrin, and the whole system can renew in 4 days' time, especially for the outer epithelium. The lower glands can be stable for a couple of months because they are much more better protected against the acid. So here you can see the parietal cells and how they work. It is very important to understand that the inside of the cell is actually very rich in potassium. And throughout the whole course, I will always come back to that. Potassium in the cell is higher than outside the cell. The sodium is higher outside the cell than inside the cell. In order to maintain that gradient, you need a lot of energy. On average, you need about 25% of your daily energy requirement, which is 500 kilocalories a day, you will need to maintain the gradient of potassium and sodium throughout the cells. So the first thing that happens is that the CO2 will combine with water. You will get bicarbonate. The bicarbonate will dissociate into proteins and bicarbonate acid. And the bicarbonate acid will go to the blood in exchange for the chlorine. So this is how the chlorine can actually enter into the cytoplasm. Then the chlorine will go out into the lumen, and it will do that in co-transport with potassium. And since potassium is very high in the cytoplasm, this is just diffusion. And so you don't need any energy to go inside the lumen. Then the chlorine is already there. The potassium comes back in, but this requires energy because it's against the gradient. And at the same time, the proton will go out in co-transport again against the gradient. In this case, you need a pump system with energy in order to achieve that. Otherwise, it will not work. So in case you have a stomach ulcer, and this means you have an itching pain somewhere in the stomach, you can go to the medical doctor and then he will give you medication to probably stop the proton pumps because you probably have too much acid production that goes into the lumen. So he gives you a proton pump inhibitor, but he's not 100% sure that it's going to work. So he will just tell you, try it out. If it doesn't work, come back in a couple of weeks. And if it doesn't work, I will give you another one. And I think he has about 5 or 6 different types of proton inhibitors. He doesn't know exactly which one is going to work, but with trial and error, he will find out which one is the most applicable for you. Okay, that's the stomach. Then we move on to the small intestine, where you have these villi, which means that you will increase the surface of the small intestine. And this is, of course, done in order to improve absorption of nutrients at the level of the GI tract. If people are malnourished, so for example, a malnourished child who doesn't have enough energy and food intake, they will probably not have development of villi. So for these children, it is not good to give them ordinary foods because they can't absorb it. In those cases, it's important that you give them specific designer foods to build up the villi again. And then when the villi are physically present again, then you can give them normal, ordinary foods. So if I look to the small intestine, then you have the villi. Inside the villi, you have blood vessels and you have lymphatic tissues. The lymphatic tissues are the green ones. The blood that is supplying foods is actually the red one. And then when it is actually full of extra nutrients, it will be blue. So this is actually the place where you have absorption from the intestine into the epithelial cell. And from there, with the blood, you can either go to the liver to metabolize it, or with the lymphatic tissue, the green ones, you can bypass the liver and go to the adipose tissue. So everything which is fat soluble will go to the lymphatic tissue, and everything which is water soluble, like for example sugars or amino acids, they will go to the blood, and from the blood they go to the liver. The absorption is done by an epithelial cell, which is an absorptive cell. A lot of mitochondria are there, so it's a cell that requires a lot of energy, and it is capable of absorbing different types of nutrients, which can then either go to the lymphatic tissue or to the blood. Here you can see a micrograph on how it looks like. The blue cells are goblet cells. They will also secrete mucus. The red circles are the nuclei from the epithelial cells, which are just layer by layer next to each other. If people are sick, malnourished, or if there is a degree of inflammation, then you will find openings here, which we call a leaky gut, because then more easier compounds can enter in the human body and they can cause inflammation and potential chronic disease patterns. The large intestine is more or less similar as the small one, but it has less villi. But for the rest, it also has the absorptive cells and the goblet cells. So also there, a lot of mucin is produced to protect the epithelial cell layers. You have microorganisms who can attach there to produce the microbiome. And you also have some enzyme activity there that can take place. But overall, it is less developed compared to what you have in the small intestine. Here you can see a little bit how it looks like. So it's a little bit a system where mainly more microorganisms can reside. Yeah, so it is fully built to, let's say, having the food for about 1 day physically there, so that the bacteria who are attached to the wall can also have interaction with the food that is physically there. With the small intestine, with the villi, you have maximum 4 hours time to absorb the food, so the transit time is much faster, and the food is also absorbed much faster than that. I will check a little bit. Okay, I will stop the recording for a minute. In case you have any questions, you can ask them. So after the anatomy of the large intestine, we go to the GALT, which is the gut-associated lymphoid tissue. So on the left side, you can see the small intestine. On the right side, you can see the large intestine. And in addition to the cells that I have already explained to you, it seems that at several places, we also have immunoreactive cells. A good example is the M cell, which is actually a transporter that brings all types of material from the GI tract inside the so-called Peyer's patches. which are just located below the M cell and which have very heavy immunoreactivity. Inside the villus, you can also find different types of immunoreactive cells, like immunoglobulins, for example, CD-type cells. So they all here the AMPs, so these are types of antimicrobial peptides, interleukins. So we have a lot of different types of immunoreactive cells that are trying to protect the GI tract against toxic or unwanted components that are physically there. And in the large intestine, we see that the mucus layers get thicker in order to protect a little bit further and also to have combinations with the bacteria that will attach to the mucus layer. And also there we have the same immunoreactive system, which is called the lymphoid follicle. A little bit different from the Peyer's patches, but the function is more or less the same. So you also have an M cell and you also have a high degree of immunoreactivity. So how do we measure that in our labs? So in our labs here, we actually try to simulate the wall, and we do that by the use of epithelial cells. So you can see the cells here. We cultivate them on a flat filter, and we place the filter in what we call a snap well, which is actually placed on a 96 well plate. And then we have two compartments. The top one is called the apical compartment. The bottom one is called the basal compartment. And then we can use all types of materials like digests, which we add to the apical compartment, and we can just look at absorption. So how much of the component is passing through and goes actually into the basal compartment. We can take samples from the apical part and the basal part, and we can just measure physically what is there. And this is a way by which we can measure transit, we can measure leaky guts, we can actually measure the impact of different types of nutrients and bioactives on the development and the stability of the GI tract wall. A second type of technology, which we also have available, but it's a little bit more complicated, is so-called organoid technology. It means that we are cultivating small organs in the lab, which are actually small intestines, which have all the cells that I just explained to you. And they're all present, not in an animal, not in humans, but just physically in the lab in a tube. And how does that work? Well, here you can see the villi, and the blue ones are the absorptive cells. The red ones are the globulate cells that produce the mucus. And then the orange ones are the hormone-producing cells, the enteroendocrine cells. So here on the bottom, you have a crypt. And the crypt is actually the birth chamber of the cells and where the cell is actually renewed. And the cell which is renewable and is called a stem cell, which is called the crypt base columnar cells. And so this is a stem cell. And next to that, the yellow ones are parent cells, which are helper cells. They are stabilizing the stem cell. So the stem cell is programmed, but is not yet differentiated. And so it's just a universal cell that can actually develop into all types of new cells with different functionalities. So the first thing that happens is that the cells will migrate up the crypt. They will go up and they come in a second zone, which is called the transit amplifying zone, where they slowly but surely start to differentiate. So to move into a new cell type. And once they go out of the crypt, which is here at this bench, the cell will fully differentiate. So the DNA at that moment will decide whether it becomes an absorptive cell, a globulate cell, or an enteroendocrine cell. And you see, you always have 5 absorptive cells, then a globulate cells. 5 absorptive cells, a globulate cells, and so on. And in this way, the cell will move up until it reaches the top of the villus, and then it will die and it will be removed, which we call anoikis. And the same happens on this side. So the cells are coming together. Here at the end, where they actually are removed from the villus. And this renewal speed is there as long as the body is well nourished. If you're very bad nourished, or if you don't have any food, so you're undernutrition, in those situations, the whole system will stop. And you don't have any renewal, no villi, and then the whole surface will become flat, and it's very difficult in those situations to absorb normal foods. So what are we doing in the lab? We are isolating the crypts. So this part we can isolate from a mouse, and this crypt is brought into a solution where we add growth factors. And this solution is producing for us the complete wall, which is then called an organoid. And it has all the cells. So you see the stem cells, the panet cells, the absorptive cells, the globulate cells, the enteroendocrine cells, even the villi. Everything is there. So we have a mini organ, a mini intestine, which we can cultivate outside the animal in the lab. The big problem with these organoids, as you can see it here, is that the lumen, so the place where the food normally is on the inside. So if we would work with an organoid, we would have to inject all our samples inside the lumen, which is quite difficult. So in order to avoid that, what we actually do is we take the organoid. In this case, for example, it comes from a pig. So you can just isolate the crypts and going into the organoids. And then we actually dissociate the organoid by the use of enzymes. And we take the whole cluster of cells and we seed them on the transwell. So you seed them again on a flat surface, but now we have all the cells in the right proportion, but again in 2D instead of 3D. And this system we can again use for normal transport kinetics, for absorption, for looking, for example, at leaky guts. So it's a little bit technical, but I hope more or less you understand how we are studying mechanisms on nutrients and bioactives in the lab today. This brings me to a first small question which you can solve with your group, but we will not discuss it in class. So it's something that you can make on yourself. The question is, mature intestinal cells in the villi have a high mitosis rate. So it means that there's a high turnover, a high renewal, and we need oxygen for that. Otherwise, it will not work. So describe to me how you can make a primary cell culture for these cells. And with primary, I mean that we isolate the cell directly from animals or humans. So it means that you have an adult cell, which you can find here. So these adult cells, for example, the blue ones, fully differentiated, they require a lot of oxygen because there's a very high renewal. How do you think that we can isolate these cells from a living animal? Yeah, try to think about a setup, an experiment that you can do to isolate these cells and to keep them active in the lab. Yeah, so come up with an idea and then think about what could be the pros and the cons of using these cells compared to using cells that you can buy, which we call commercial cells, or cells that are grown in organoid models. So organoids, I just explained, cell lines are something that you can buy commercially. And here we try to isolate the cells ourselves directly from an animal. So try to think a little bit what could be the pros and the cons of using this technique of primary cell culture isolation. Good, and this brings me to the last part of the lecture, which is on motility. So about movements. So movements that can happen in the GI tract. We call them peristaltis, which means alternative contraction and expansion. Segmentation, which means rhythmic contraction and expansion. And then commutative movement, which means the peristaltis and the segmentation at the same time. There are two examples of peristaltis and contractions that are important for you. The first one has to do with the stomach, and we call that adaptive relaxation. So you should try to understand what this process means. I have two curves, one on the top and one on the bottom. In your course notes, everything is translated, so you can look at it there, but I see that it is here in Dutch. So here on the x-axis, you have time in minutes. And here at 0 time, you give food to a person. You will see that when people are consuming food, the food enters into the stomach, and then the pressure in the stomach will increase, which is this line. At a certain moment, here, after 7 minutes of eating, the pressure is so high that you will stop eating, because you get a feedback that goes to the brain that says the stomach is full, I have to stop eating. If you don't have adaptive relaxation, it means that after 7 minutes of food intake, you will stop eating. However, in reality, when the food comes in the stomach, it seems that the stomach wall will relax. So it means that the volume of the stomach will increase and the pressure will stay low, as you can see here. So you can keep on consuming food until 15 minutes. And then after 15 minutes, the stomach is really full. And then you have an exponential increase of the pressure, meaning that you will stop eating. Yeah, so in this case, adaptive relaxation helps you to consume a little bit more food. Now think a little bit yourself how we can use the process of adaptive relaxation to reduce food intake in humans. So what can you do in order for people to eat less by influencing the process of adaptive relaxation? Yeah, so how can you do that? Think a little bit about it. The second process I want to show you is peristaltic contraction, which takes place also in the stomach. So when the food is entering in the stomach, as you can see here, there is a peristaltis that goes from the top to the bottom. And this is the red arrows that you can see. So they actually push all the food forwards until it reaches here this pylorus, as we call it. So it's actually a vessel or a tube where you can actually go into the small intestine. And what actually happens is that this vessel will close, the pylorus, and then the food will actually be squeezed in a very small area. Only the very, very small particles will pass through. The bigger particles will flow back. And you can see that also here, all the bigger particles will flow back. The smaller particles will pass through. And once they flow back, they have sort of an expansion, so they are just destabilized and they become smaller particles. And then the whole system starts again. Yeah, so the stomach and the peristaltis is actually intended to control the supply of food to the small intestine, but also to reduce the particle size in such a way that you have a homogeneous supply of particles to the small intestine. And you don't have any overload there, and then normally the absorption can take place in the small intestine. Here you can see stomach emptying. So this is the time. This is a liquid like water. This is a porridge, and this is a pancake, so a solid type of food. So by playing around with the texture of the food, you can also influence the residence time in the stomach. Yeah, so think very careful if we give you some questions about residence times. Think about the techniques that you can use to increase residence time or to reduce it. By just playing around, for example, with the texture of the food, you can already change the residence times. How can we measure that? So peristaltis and residence times nowadays are measured by the use of stable isotopes, not by radioactive isotopes, because radioactivity has been used in the previous century. but today it's not used anymore. Not with animals, also not with humans. We are mainly working with stable isotopes who are not radioactive, but which are sufficient to distinguish them from the normal molecules. So people just consume a small tablet that contains carbon-13. It is digested in the stomach. And what you do is you take samples from the breath, because after the stomach, the stable isotope is absorbed. It goes from the blood to the lungs, and from the lungs, it is expired in the form of CO2. So you can just measure the release of your isotope by the lungs, and this release is directly correlated to the residence time in the stomach. Yeah, so there's a direct correlation between residence time and release of the tracer by the breath. A remaining question for the second group, so task B. There's a hormone which is called gastric inhibitory peptides. And this is a hormone that can reduce the activity of the stomach. So this hormone is released on the basis of the composition of the intestine. So I already told you about the taste receptors. We can actually measure what is physically present in the intestine. And based on that, we can release GIP. And GIP is actually reducing the activity of the stomach. So my question, explain on the basis of a fatty meal how the body knows when GIP needs to be released. So think a little bit how a fatty meal will be digested in the stomach, and then try to think about which components are going to trigger the release of GIP by the stomach so that the stomach can stop working. So how does the mechanism work? And this is my final slide. There is an article, you can find it on the website at Euphora. It's produced by Gorevsky et al. From 2022, so it was published last year. It's a very recent technology. You can also see movies. I think there are 6 movies in that article that show you how the system works in a dynamic way. I think it's a fascinating article that explains to you how far we already are with designing and developing model systems in the lab. And I will just explain one slide, so this one. So here you can see what we call a scaffold, which is actually the villi. Remember the villi that I explained to you with the crypts? And then here you have the size and the shape of the villi. This is how it is normally in the small intestine. This system has been reproduced in the lab by using a sort of a plastic cover. They just produce with 3D printing, you can do that. You can just produce the same shape as what normally happens in the small intestine. And here you can see how it looks like from the top. So these are all the villi, so these plastic materials that have the same shape as in a human body. And here you can see it from the side. So here are the villi, and here are the crypts. Yeah, so here at the bottom is the crypts, and here are the villi. So this surface is now used to produce a surface layer of cells. And we isolate the stem cells in the same way as I explained to you before. So you start from an animal, a mouse. You isolate the stem cell from the wall of the animal, and then you inject the stem cells here in this small crypt layer, which is actually done here. So they just inject it here in the crypts. And then you just wait. You incubate it at 37 degrees Celsius, and then you see that the cells start producing and multiplying. And more cells are there, so the cells come out of the crypts. And then it seems that these cells will creep up the villus until they reach the top. And at the top, they are just disappearing and they are dying off. So exactly the same process as what really happens in humans. And here I have the proof that this is correct, because the green color are actually the markers which were used to identify the stem cells. So you can really see that they are physically there in the crypts. If you go to the red colors or the purple colors, these are the fully differentiated cells. So the cells that you will normally only find here at the top of the crypts. And so this system means that the stem cells develop, creep up here the villus, and at the end, they just go off and they die. So the full renewal of the wall can also already be simulated now in an in vitro system in the lab, which is a very powerful new technology that we have. And so this is the end of this lecture. I will stop the recording here. So if you leave us here, I would like to thank you for your attention. I will stay here for another 10 minutes. For everybody of you who still has questions on what we discussed today, you can ask them after stopping the recording. So thank you and have a nice day.